So this video is going to focus on nucleic acids, but DNA, and looking at the overall structure of it. When we ended on the last video, I was talking about the basic nucleotides and nucleic acids in general. And I told you that the important part that we've been talking about are these bases, that these pyrimidines, which is cytosine, thymine, and uracil, cytosine, thymine, and uracil, and then these purines, which is adenine and guanine. And we know the name changes when we put them onto a sugar. And we're going to focus on DNA in this section. So when we think of B, this is DNA. I'm going to show you, similar to the way we talk about proteins, we're going to talk about primary, secondary, tertiary, and quaternary structure. So let's start with the primary structure. And as it is with amino acids, primary structure is the order of the nucleic acids, or the order of the nucleotides. We're talking about DNA here, so when we're talking about DNA, the sugar that we're talking about is deoxyribose. So, and the nucleotides, that it's deoxyribose, and then our bases are the A, T, C, and G, not uracil in this case. And so when we think about the order, typically we have a 5' and a 3' end. And remember, I told you that generically last time, that if we have a 5' end, and I'm going to draw this cartoonishly, and I'll draw a few of these together. We have this 5' end, and the 5' end typically has a phosphate on it, like this. Okay, so this would be what we call the 5' end of a piece of DNA. This is a single nucleotide. Doesn't matter what base I put, I'll just put A here. And then we don't have an oxygen on this 2' here, and then this is the 3', and we would have another phosphate. Okay, and we would have then another, and I'm running out of room, let's put a T here. And then I'll put an OH here. This is the 3'. And we just keep going. We do this. I put an OH here. This is just 2 nucleotides, but I'd keep going. And you'll notice when we write these, that would be negative. A couple things that you should realize is that we typically have a phosphate. And then we have a sugar, which in our case is deoxyribose. And this is the 5' end. And then we'd have a phosphate. And then we'd have a sugar. And then we'd have a phosphate, and then we have a sugar, and then this would have a 3' end that's got an OH, a free OH on it, right, like that, okay? And then off of each of these sugars, we'd have a base, and off of that base would either be A, T, C, or G. Off this one, A, T, C, or G. And same thing, A, T, C, or G. It doesn't matter which one we have. But in this case, I just drew 2 together, and you can see that the phosphate, sugar, and it would just keep going. And then this base would sort of be off to the side. So that's basically what and I'd want you to be able to draw this. I know this is going to be tricky in an online situation, but you already know how to draw one of these because we have drawn ATP and AMP, even though that was ribose in that. This is deoxyribose that we're talking about. So that's the primary structure. I'm going to show you a quick slide of this. So if you look, let's make sure this is what we want. AT, that is RNA. Okay, I know this is DNA. I always know the DNA because it doesn't have an OH group on it. And so you can see this would be the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and then the bases would be linked off like this, and this would be T, G, C, and A in that order. So 5' end has that. phosphate, the 3' end doesn't. One thing you'll notice, they made a mistake here. That should be a little negative on that. But one thing you should be aware of is that DNA overall is negatively charged. You can see all these negatives, and that's going to be important when we kind of move forward. So that's the first thing. That's primary structure. And then secondary structure. So secondary structure, basically with DNA, remember we're talking about DNA, is two single-stranded DNA chains running what we call anti-parallel. So I'm going to show you how we draw this first. When you think of 5' whatever, 5' going to 3', anti-parallel means that we have another strand going 3' to 5'. And typically when we draw these, that's what anti-parallel means. One's running one way, the other's running the other way. And when we connect these, and like I said, we're going to be kind of doing some cartoony stuff here. If we had A, C, T, and G, and that was the 5' and that was the 3', the anti-parallel chain, the T binds with the A, the G binds with the C, and same thing over here. And they'd run like that. And that's a cartoon of that, but that's what they look. And we know, and we'll talk a little bit more about this now, hydrogen bonding actually helps stabilize this. So when you have an A and this anti-parallel, it actually has two hydrogen bonds that it connects with the T, and C's actually have 3 hydrogen bonds when they connect with G's. So I want you to know this. This is stronger because it has 3 hydrogen bonds as opposed to 2. And so what you see in a lot of regions of DNA where it opens up a little easier, because you have to open up DNA to duplicate it, you have to replicate it, you have to open up DNA in order to make a protein out of it, or a piece of RNA, and so you'll see these repeats, and they call them Tata boxes, and they're actually regions of the DNA that I guess open easier. And it's because they have fewer double bond or fewer hydrogen bonds that are connecting them. So let me give you a quick-ish picture of this. This is kind of neat to see. I think DNA is just fascinating. If you look, this is adenine and thymine. There's 2 double bonds between them. And then guanine and cytosine, you can see there's 3 hydrogen bonds. I said double bonds. I meant hydrogen bonds between them. And that's actually what helps stabilize that structure. It isn't the primary thing that holds this together. We'll get into that here in a minute. But I want you to understand that that is what's actually taking place. So these hydrogen bonds, I'll write this on the next page, hydrogen bonding helps to stabilize the secondary structure. All right. So know that they run anti-parallel, and that C binds with G and A binds with T. All right. So, and typically if you just see something like A, T, C, G, A, G, T, it's assumed that this is a 5' end and this is a 3' end. So when you see something written like that, that's what they're talking about. And they typically don't write both strands because they know you can draw the complementary strand is what we call them. And I'll define all that stuff later. The next one is tertiary. And tertiary structure is basically the DNA helix arrangement. And you might say, well, there is a DNA helix. Well, there is. There are multiple forms of this tertiary structure that exists. And I'm going to talk about 3 that I want you to know. But the most common, it's the helix, and there's a couple different. And right when you see them, you'll understand what's going on. because it's kind of like, well, it's sort of intuitive, like, yeah, it has to be that way. Well, it's sort of intuitive, like, yeah, it has to be that way. So when we see DNA helix arrangement, I'm not going to be able to draw these very well. I am going to attempt it, though. The most common tertiary form of DNA is what's called bDNA. And bDNA, I'll define it for you. bDNA, it's a tertiary structure, and it's really the principal form of DNA. This is how we typically find it, okay? And so the way it looks, it's what we call a right-handed helix. We won't get into what that means, but there's a right-hand rule that it curves kind of the way if you use your right hand and curve your hand around, it curves and goes in the direction of your thumb. So if we were to draw kind of a generic double helix, let's say I'm going to draw it a little bit tighter on one side and a little bit bigger over here. What you have is you form, when you do this double helix, and I know that's a beauty right there, you form what's like a big groove and a small groove. So there's what we call a major groove, and I'll obviously show you a better picture of this because this is embarrassing, but and then there's a smaller portion of it, like over here, that we call a minor groove. And what's neat about it is proteins actually recognize these larger or minor, the larger and smaller minor grooves. The distance for this is about 22 angstroms, and the distance for this one is about 12 angstroms, okay? And so overall, a single turn of the DNA is about 34 angstroms to get from one turn. So a single turn is about 34, I can do math, 12 plus 22, 34 angstroms, which is the 12 plus the 22 angstroms long. And then it's about 10 bases long. So about 10 nucleotides that make it turn like that. Okay, so that's the kind of the basic structure of bDNA. I'll show you a good image of it. I do want you to know that, like I had said this, but DNA in general is negatively charged. Due to the phosphates, right? They all have that negative charge on them. And so as a result of this, whenever you have DNA, it is littered with sodium and magnesium and positively charged proteins, because they're attracted to the negative charge of the backbone of that sugar phosphate that keeps going down. There's all these negative charges, so you get these things that are oppositely charged that want to be around it. So let me show you a bDNA. This is a better image of it, and you can see that there is kind of a major groove up here and then a minor groove, and they run anti-parallel with one another. And even this is a cartoon, right? And it's got the negative charges on it, but it gives a better picture of what I was drawing, okay? And then I'm going to tell you the other two, and then I'll show you an even cooler image of all three of them together. There also is what's called aDNA, and this is a right-handed structure of DNA, and it is really, it just has different dimensions than the bDNA, but it occurs when you have a DNA and RNA hybrid. And if you think about this, this is really important. When you think of replication and it's occurring, and you remember those Okazaki fragments, they're layers of DNA that are laid down to help prime and make the thing replicate. And if you've never heard of that, you will by the end of this section. But your body needs to somehow know that there's RNA in your DNA, so it comes along, and because it's in this aDNA structure, which is a little different, it distorts the DNA, so it knows that it can come along and cut that out. But the OH of the the RNA, because remember, RNA has ribose, not deoxyribose, distorts the structure. Okay, so it's this right-handed structure that has a little bit different because of that extra O that's there. And then the last one I want you to know is what's called zDNA. And zDNA occurs when there's a bunch of CG repeats in the DNA. And this actually is a left-handed structure. All right. So let me show you a good image of this. These are the different strands. And so bDNA, this is the one that we kind of think of. You know, it sort of has a major groove and a minor groove, major, minor. aDNA, it's a little wider, and you can see that its shape is different enough that a protein coming along would go, oh, something's up with this. And what's beautiful about this bDNA is if you look at these rungs, these bases that are attracted, they're really stacked nicely on top of one another, even with some of the others, but this one just has kind of an elegant structure. And then the zDNA is a little different structure. Okay, so when we think about all this DNA secondary structure and these tertiary structure that exists, both the overall tertiary structure is stabilized not only by hydrogen bonding, but it also is stabilized by hydrophobic stacking of the bases. Okay, so the tertiary structure is stabilized by hydrogen bonding, which I already wrote. Between the bases, the A, C, T's, and G's, and hydrophobic stacking of the bases. And if you go back and look at these structures, it's because you get, if you go back and look at just the bases here, you can see there's a bunch of pi bonds that are in these structures, and when they stack on top of one another, they have they induce dipoles in these pi bonds, and it actually helps stabilize that overall structure. So I want you to know that it's not just the hydrogen bonding, it's also the hydrophobic stacking. And your book has cartoon images of this. Like here are the bases stacked, and you can see there's little cartoons like they're, structurally. But those are important just to understand that those two are like that. Now, I also want you to know how all of this DNA fits inside the cell. And so you've heard of this before. You know, if you were to string all the DNA together, it would go, extremely, some ridiculously distance, that was overwhelming. That's all squeezed into this cell. So the question is, how does all the DNA, the billions of bases, fit inside the cell? Well, it does it because of something called supercoiling. And I wish I was in class. Maybe I'll make a video of this, but whenever I try and get a extension cord out of my garage to do something outside, I realize that someone has supercoiled the extension cord because they wound it up around their arm, and it introduces all these twists, and then when you try and untwist it, it takes forever, and it's because of supercoiling. So maybe that doesn't frustrate anybody else, but so think about it in terms of DNA. So it's basically adding twists to consolidate DNA. And if you were to take like a rope or something, and you had a rope, and bacterial DNA is cyclical like this, if you were to turn one side of this, if you cut it and turn one side, it actually turns into an 8, and then you cut it and pull it apart and twist it again, put another twist in it, it ends up looking like this. And then you keep doing this, and this is what the concept of supercoiling is. And you can do this in non-cyclical DNA too, because if it's long enough, it'll start curling up. Okay, so I want you to know the difference between underwinding this is where you put what's called a negative supercoil. And this unwinds DNA. And we think about that as really opening the strand. And then there's overwinding. And this is what's called a positive supercoil. And this winds the DNA and kind of tightens it. I don't think of opening it, it kind of tightens it. So we have enzymes that do this, that underwind and overwind, and they're called topoisomerases. And they are enzymes that cause supercoiling. Okay? And so they have a different way. There are different types that we don't care that much about, but they can either break 1 or 2 strands. So it can just do 1 or 2 when doing the supercoiling. And typically, because of that, they use ATP to make this happen. Okay, they consume ATP. It's something that normally wouldn't take place. So, so like I said, it doesn't have to be cyclical DNA. Both eukaryotes and prokaryotes still supercoil their DNA. And then you think, well, once you have all this supercoiled DNA, we have what are called histones. And these are proteins that are rich in lysine and arginine, which you know what lysine is. You probably haven't memorized arginine. That help. So rich in proteins that are rich in lysine and arginine that help stabilize the supercoiled DNA. All right. So you can, I think I have an image of this. This is an example of supercoiling I have here. Whether it's a single, you twist it, starts curling up. Here you can see you can start twisting it and coiling it up. And then you get these histones. Well, that's the gyrase that's actually putting the things in. This is a slide I was looking for. These histones that actually the DNA ends up wrapping around. Okay. So that's what I want you to know with that component. And then so primary, secondary, tertiary, we got this wound DNA, these histones. By the way, you might want to make yourself a note. These are positive. That's why they're attracted to these. Why are they rich in lysine and arginines? Because those positive charges attract the negative DNA. Okay, that's important. And then the last one is quaternary structure. And this is basically DNA interacting, and maybe I got ahead of myself here, interacting with other proteins. So things like your histones, probably I should have put things like histones, other pieces of DNA, other pieces of RNA interacting. Ultimately, they can make those structures. So you've probably heard of them as things like chromatin, which are basically DNA and protein C complexes. And then there are terms like nucleosomes. It's like one wrap of DNA around a histone. And I don't care. I'm not going to ask you questions on these. I just want you to know that that's where those terms come from. They're examples of quaternary structures, okay? And I showed you an image already of those. Now, the last thing in this section I'm going to talk about with DNA is that you can denature DNA, okay? So and that's going to be extremely important for something we're going to talk about. So you can denature DNA, just like you can denature proteins, just like you can denature enzymes. So what does it mean to denature? But this is what's cool about this. This is obviously a temperature-dependent process, okay? It is dependent on how much G's and C's. So if we're doing experiments with smaller pieces of DNA or bigger pieces, we care about the GC content. Why? Because they're tighter, more tightly held. So it typically depends on the GC content. Now, you know, in your body, you maintain a given temperature. This is in the lab. If we're going to do something with DNA, we can denature it. Your body knows how to open up and close DNA without denaturing the whole thing. So this is sort of an experimental thing that we do in the lab if we wanted to denature. It also is called the denaturation process. Is also called DNA melting, which I don't love, but it is what it is. So one of the things that I'm going to draw this on the next page, the way we would explain this is, let's say we are going to draw a graph and we're going to put temperature here. Now the question is, what do we put on this side so that we know what it means, whether it's denatured or not? Well, interestingly, we used absorbance. And so we use absorbance at 260 nanometers. And if you remember, that's outside the range of the visible, 450 to 700-ish. This is actually UV light. Okay. And what's interesting is double-stranded, DS, double-stranded, I'm going to write that down, I'm going to use that a lot, stranded DNA absorbs less UV light, and then SS DNA, which is single-stranded or denatured, right, absorbs more UV. And I'm talking about this particular frequency of 260. And the reason that is true is because it's those purines and pyrimidines that are picking up and absorbing that UV. And in a double helix, that's sort of, I guess, shielded from absorbing the light. So we measure this of the absorbance of 260 nanometers. And so if you have low temperature, you have double-stranded DNA. And as you increase the temperature, it ends up looking like this, okay? Now, if you've taken a calculus class before, this is what's called an inflection point. You don't have to know what an inflection point is, but you do have to know where it switches, where it kind of goes from curving up to curving down. This is what we call the TM, which is the melting point. And you'll notice it doesn't all happen at once. So if we come down here, this is the actual TM value. It's sort of a range, but it's double-stranded here, and up here it's single-stranded, and it kind of starts taking place here and here. So it's a melting range that typically, and then we center it on that inflection point, but it's not all double and all single in that it's kind of stuck together a little bit. So if you think raising the temperature gives you more single-stranded, lowering the temperature gives you more double-stranded. And the nice thing about this is if you lower it slowly enough, so if you melt your DNA, you single-stranded, and you go down slowly enough, it'll go back perfectly, unlike proteins sometimes. So I do want you to know that if the temperature is lowered slow enough, the DNA will perfectly go from single-stranded DNA to double-stranded DNA. All right? So I included a slide here. So you think of double-stranded absorbance. You raise the temperature, it starts to melt, unwinds, get your inflection point, and then up here after this range it's completely, but we call that transition temperature, that TM, that melting temperature, right there in the middle. Okay?